All right, hi for everyone that hasn't met me. My name is Hanna, and I'm a graduate student working with Doctors Bramlett and Dietrich here at the Miami Project. And my work really focuses on recent advances in machine learning and how these may fit into the neurotrauma laboratory. So, briefly, machine learning is a subset of artificial intelligence, or AI. Now, I'm sure we're all familiar to some degree with AI, as these systems have actually integrated themselves into our lives in a number of ways, from self-driving cars, like with Uber, or even Siri. And AI is really just a thinking machine that is able to complete tasks in a way that is reminiscent of the way that humans might complete these tasks, and then possibly even interacting with its environment and learning from new cases. Machine learning is the part of artificial intelligence that really gives the machine the ability to learn from data and make predictions about future cases. Now, machine learning itself can be further broken down into subcategories, and the two that we are going to be focused mostly on today are supervised and unsupervised learning. Now, these two categories differ in whether or not the machine, when asked to analyze a data set or complete a task, is provided information about the target goal. For example, say I have a series of pictures of cats and dogs. If I just give the machine these pictures and say, analyze them without any information about what I'm looking for, it will cluster them by similarities it finds, whether they be patterns or correlations. And this would be unsupervised. Now, if I tell the machine what I'm looking for, say cat or not cat, and then give it a series of examples or a training set where it can learn what a cat is and then test its ability on examples that it hadn't seen before, then this would be supervised learning. And we'll go into some more detail about these techniques a little bit later in the talk. However, machine learning in general has already been a pretty hot topic in medicine in recent years, given its ability to integrate huge amounts of data in ways that is well beyond human capabilities. Now, these techniques, when developed correctly and with strong data sets, have the ability to accomplish some really powerful goals, such as driving us towards more personalized medicine or increasing physician efficiency. However, these upsides are not without caution because machine learning can suffer and how easy that they are to interpret, and this can lead to things like bias. And this has actually been a problem in other applications of machine learning, such as facial recognition, where the actual algorithms were designed on a data set that did not equally distribute samples across races and genders. And then it introduced levels of bias that were actually harmful in its applications. Additionally, and this is becoming more and more problematic in our fields, is that with the increased interest in these different techniques, um, some of the work is not properly developed in a lot of ways, and we're seeing a lot of very weak papers published because in many cases the reviewers just don't know what to look for. And basically, if your data or your application of these techniques is garbage, your results are going to be the same. And lastly, a topic that applies heavily to the clinic, there isn't yet a way to standardize who's at fault when one of these machines makes a mistake. And that can be a major hurdle for fully integrating these techniques into the medical fields. But there have been numerous success stories. Probably the most well-known application of machine learning is its ability to outperform humans actually in identifying pathologies or damage on images generated from things like MRIs or CT scans. It has also successfully been able to predict treatment toxicity in cases of cancer treatment. And we see it a lot in the preclinical or experimental side of things for dimensionality reduction. That is, you have different techniques such as genetic sequencing that is generating huge amounts of data, and these techniques actually have the ability to make those data sets a little bit more interpretable. Now, the main topic of my research is traumatic brain injury, or TBIs. And to understand how these techniques might fit into brain injury research, we need to have a sense of the current state of TBI. Now, TBIs are a significant healthcare concern that affect nearly 1.7 million new Americans each year. Now, that 1.7 million is actually 6 times the incidence of MS, breast cancer, HIV/AIDS, and spinal cord injury combined. And given that these injuries can lead to lasting deficits, it's now estimated by the CDC that over 5.3 million Americans are living with TBI related disabilities. And despite this alarming incidence, research has ultimately failed to identify effective treatment strategies for TBI patients. And this isn't because the researchers are bad or there's just not any work on it. There's actually been 40,000 papers published on TBI, and we know quite a bit about the pathomechanisms that underlie the deficits following TBI. Theoretically, this should mean that we have good therapeutic targets, but effective therapies are still difficult to identify. And the answer to why this is ultimately lies with the complexity of this disease. TBI isn't a one hit and done kind of situation. Following the mechanical insult that results in a disruption of the underlying neural tissue, there are a series of pathobiological cascades that lead to further cell death and dysfunction for weeks to months after injury, and then give rise to these chronic neurological deficits that the patient experiences. Furthermore, that heterogeneity doesn't stop with injury type, it also changes with where on the head the person was impacted, it changes with the severity, and even pre-existing factors such as were you drunk when you hit your head can impact how well a person recovers from these injuries. Now, we and others are starting to ask the question of whether these differences in the TBI population are actually one of the driving factors for why we've had so much trouble in identifying therapies. That is, should an injury caused by impact be treated the same as one caused by a blast or a shockwave? And these individual variabilities, however, become extremely difficult to analyze with the currently used analytics, which does open a door for some more computational techniques such as machine learning. And in fact, it is actually the case that therapies tested for their effectiveness following brain injury are failing or even deleterious in some cases, yet they're working for other patients. And this is true both in the laboratory setting and in the clinic, just further highlighting the need to capture some of the individual variabilities that exist in TBI data. So then we can ask the question of what role does machine learning play? And major sweeping questions from the TBI field have been addressed, such as will the patient survive? How long will they be in the hospital? Will they have an abnormal CT scan? However, some more intricate questions, such as degree of recovery or optimal treatment, have yet to be well hashed out. So with that, let's turn our attention to how TBI studies are typically analyzed, or the data that they generate are typically analyzed. Now, I'm sure many of these techniques are very familiar to most of you, but I'd like to point out that univariate techniques are, as their name implies, focused on one variable at a time. That is, you can analyze something like cortical lesion volume or performance on a learning and memory task. However, you lose some of the correlations and patterns that exist between variables. And this is important to consider when we're looking at functional recovery as a whole rather than as a function of individual metrics. Multivariate statistics, on the other hand, draw in techniques from fields like linear algebra in a way that do allow us to analyze matrices as a whole with multiple subjects and multiple metrics for each subject. You can think of your Excel data sheets full of rows and columns and analyzing that as one test versus univariate setting where you would have a statistical test for each column on that data set. Now, a number of these techniques, such as principal component analysis or some of the clustering techniques, are becoming more and more commonplace in biomedical research. So I'll go into further detail about those here in a moment. But briefly, while in some cases they are pretty synonymous, machine learning and multivariate statistics do differ in their purpose. That is, multivariate statistics are aimed at providing the scientist statistical inference or interpretability about their data set in a way that makes their data more understandable. On the other hand, machine learning really puts its focus on predictive power of its models, and this can lead to a sacrifice in terms of how interpretable those models are. In other words, they're similar in many ways, but they do quite obviously differ based on how techniques from other fields might be incorporated. So then, the question becomes, what can machine learning actually teach us? And the answer to that really lies with your field and with your goals. Someone in, say, business might be interested in these techniques for their implications in advertising or market forecasting, while we in the medical field are obviously interested in answering questions ranging from image classification to big data visualization, and of course, diagnostics. And like I said, we're seeing a number of these multivariate and machine learning techniques showing up in the medical literature. And PCA is a prime example of this. PCA is essentially a way to reduce the dimensionality of a large data set in a way that makes it more interpretable while minimizing the information lost by reducing the dimensionality. And it does this by creating a series of components, known as principal components, that identify a combination of the original dataset features that contribute to the max amount of variance in the dataset. You can then use these techniques to gain a greater understanding of what these variables might be, or as a pre-processing step where these principal components actually become your training features in a machine learning model. And I'll show you examples of both of these applications towards the end when we look at my own research. A second technique known as TSNE has also shown up a number of times, even in these Miami project seminars. And briefly, TSNE is a dimensionality reduction algorithm able to cluster samples based on probability distributions. This is useful in, say, something like cell sorting, where you aim to identify cell type by gene expression. And then each of these clusters would correspond to a different cell type. A word of caution though, TSNE is very sensitive when it comes to the predefined parameters, and it needs to be validated because it can find clusters where clusters don't actually exist. However, almost all of the work that has been done with these techniques and with some of the other ones have been with large clinical or sequencing data sets due to the fact that they typically require huge amounts of quality data to train a useful intelligent system. Transforming these techniques into a form that would be useful in the laboratory, where animal subjects are typically very limited in number, requires insightful ways to generate and process the data. And these are some of the major hurdles that I aimed to address with my dissertation work. And essentially, I'm asking the question of whether or not we have the ability to apply these techniques in a way that will better inform preclinical or experimental research. As a brief example, you can consider the infinite number of combinations of therapies we could test for TBI. If we establish a workflow that's effective in addressing the hurdles of limited animal data, we can potentially use these techniques to help identify which combinations of therapies would have a high potential for additive effects. And clearly, this would save enormous amounts of time and resources that would otherwise be spent in brute force testing. So then, what does using these techniques actually look like in our field? First, we have to acknowledge that what types of data we have in our data sets, because different machine learning models are better suited for the analysis of different types of data. We then have to move into the pre-processing phase, which is rather extensive for these techniques, because you really have to teach the machine what you as a scientist knows. For example, a scientist trained on, say, a behavioral task would be able to look at the data and right off the bat say, wow, that rat sucks, or that they obviously didn't learn anything. Whereas a machine would just see the number, Without context, it's just a number. So keeping this in mind, we have to really devise the best way to represent each feature relative to both itself and the other features in the data set in a way that not only informs the machine of what we know, but does not introduce experimenter bias. We then have to shape our research question as a machine learning task or as a combination of machine learning tasks to get to the answer we are looking for. So now to get a sense of what my data set actually looks like, let's run through how TBI is modeled in the lab. There are many different devices that model different injury types, like the ones I mentioned a little bit ago. And two of the devices I have pictured here. On the left is the Controlled Cortical Impact Device, or CCI, which causes its injury by quickly lowering an impactor tip a few millimeters onto an exposed dura and results in a mechanical disruption of the underlying tissue. This creates a very severe focal injury at the point of that impactor tip. The fluid percussion device, on the other hand, also results in a mechanical disruption of exposed dura. However, it does so with a fluid pulse. And this results in a less severe focal, but also a diffuse injury of the brain. Functional recovery following administering these injuries is then measured with a number of different quantifiable assessments that are designed to assess things like motor or learning deficits after injury. These tasks often generate data sets that look something like this spreadsheet with a number of columns for the metrics that were measured during the task. And then these data are typically analyzed using those conventional univariate techniques such as ANOVAs and generate tasks like, or graphs like this. In an attempt to set ourselves up for the best chance of success in applying machine learning to the TBI laboratory data, we accessed the rather large animal data set collected by the consortium study known as Operation Brain Trauma Therapy, or OBTT. Now, OBTT noted in its study design that many labs are using different devices, so like that CCI versus the FPI device, and effectively modeling different types of injury. And that this may be contributing to some of that lack of translatability we've seen from bench to bedside in terms of finding effective therapies. To this end, they hypothesized that if they could find a therapy effective across three very different injury types, then it may be best suited for a successful translation to the clinic. To test this, they identified 12 different therapies that had previously been reported in the literature as beneficial, and then tested them across a number of different functional recovery metrics, which included physiology, biomarkers, motor, and cognition. They also had histology, but I did not analyze that in my studies. Now, in this particular study, they utilized a scoring system or a scoring matrix that addressed each therapy's performance on these listed metrics and analyzed them for significance with the typical univariate statistics. What I want you to take away from this slide is on this matrix, the top score was actually 66. And the best performing therapy was levetiracetam, also known as Keppra, and it got a 10. Now, I don't know about you all, but if I got a 10 out of a 66 on a test, I was definitely hiding that in the bottom of my book bag, hoping that my mom never saw it, or else, well, I probably wouldn't enjoy the outcome. Anyway, while these results were far from groundbreaking, these data are clean, complete, and tabular, and it's rather large in terms of animal data sets go. So these are all checkpoints for the application of computational techniques, and we were pretty excited to see what kind of novel insights we could get from reanalyzing some of these data. So to dive right in, I modified this slide directly from one of the OBTT presentations, but it does give you a little bit more background regarding the treatments that we were analyzing in our studies. So we chose 5 treatments that covered a wide variety of therapeutic targets, and we tested the therapies that had 2 doses per pharmacotherapy, with the exception of minocycline, which only had 1. We then move forward with a pretty typical exploratory data analysis and moved right into the feature engineering steps for our multivariate analyses. Now, like I said at the beginning, if you feed your machine learning system garbage, it's going to give you the same. So we had to engineer our raw data in, like I was talking about a moment ago, the way that informed the machine of what we as scientists know. So here you can see we included all 4 feature groups, metrics from those 4 feature groups, so individual metrics, and then engineered them in a way that not only informed the machine of things like change over time as opposed to raw data, but also normalized all of our metrics so there weren't any scaling discrepancies, which would introduce some sort of arbitrary bias. Now, one thing to keep in mind, these are the data for the FPI animals, and this is going to change slightly when we move into the CCI model. So we're going to have to compare and contrast both in terms of the features that are included and in terms of how the animals are recovering differently depending on the injury that they sustained. Now, our first somewhat exploratory question became, can a machine discover novel insights from data that were previously deemed insignificant? Now, I have a couple of metrics pictured here, but it was true across the board that nothing in the minocycline data was reaching statistical significance. And so we applied a combination of dimensionality reduction, which would have been the t-SNE technique that I talked about a little bit ago, and k-means clustering to determine if there were patterns and correlations present within the data set that would allow the machine to cluster these subjects, represented by individual dots here, by the treatment they received, despite giving no information about the treatment groups. Now, with minocycline in red and vehicle in gray, we actually found this to be the case. And while it's far from perfect and that some of the subjects were misidentified, we were confident the machine was uncovering treatment effects that were missed by the initial analyses, and that supervised learning classifiers may drive or discover further insights from these data. So we moved towards supervised classification with the same curated data set, and we had to decide what our target was going to be. Like I described earlier, you can have a series of pictures, ask the machine to identify cats, and that would make your output options cat or not cat. But as you can imagine, that output is a little bit more complicated when you're talking about the medical fields. And admittedly, in our first study, we went big, go big or go home, right? And we attempted to define recovery. Can we predict recovery? However, defining what recovery is a very open-ended question. And it becomes further complicated when we're speaking within the frame of animal research. And even reading here, you can start to understand how we're introducing experimenter-defined parameters and possible bias. And that really kind of takes us away from one of the true powers of machine learning, is that it steps away from hypothesis testing and automates some things in a way that removes some of these biases from statistical analyses. But aware of these concerns, we move forward. And we tried to limit the bias by just focusing on individual end study metrics and their distribution across histograms. We then chose the most evenly distributed metric that was at the end of our study, so 30 days, about a month after the injury had been sustained, and called the samples that ended in the leftmost bin not recovered, and the samples that ended in the rightmost bin recovered. Now, the metric that happened to be the most evenly distributed was the memory retention task, and some of you may be familiar with the Morris water maze. This was the probe trial from that Morris water maze. And then we framed our research question as, can a classifier predict this recovery output from a metric recorded a full month after injury, given training on metrics recorded during the first week of recovery? To frame this as a machine learning question, we then had to choose classifiers we are going to train. And the answer to this usually isn't one classifier versus another, it's actually many classifiers. And you can imagine if you and me or all of us were assigned a task for how to get from my house to campus, we could all come up with very different ways to do that. I may drive and make a wrong turn, someone else may take a helicopter. One answer is not necessarily better than another, and they all typically accomplish the same goal. Classifiers are similar in that they're all trying to accomplish the same task that you give them, but they do so in different ways. So I'll briefly run through some of the strategies that each of these models uses to give you a general idea of what's going on behind the scenes. So let's look at decision trees first. Now, when we're talking about decision trees, imagine a flow chart where the separation points are formed and determined by something called information gain. Now, information gain is looking for the breakpoint that would lead to the biggest separation in samples. So say we had 10 samples and there was a breakpoint that could be made that could separate 5 samples from 5. This would be a greater information gain than just separating out one sample from the 9. However, it can separate out one sample from 9 if that's the best it happens to find. And you can see how this becomes a little bit overfitted in the sense that if we pick out one sample by one sample by one sample, that's not going to be super generalizable to new cases. And we're really trying to predict something that can be applicable to new cases. And to get around this a little bit, you can actually make an ensemble of these decision trees known as a random forest. Now, a random forest is going to be a bunch of these flowchart style trees, and then the average vote of these trees becomes the output class. This is not as prone to overfitting, however, it is much harder to interpret how the machine got from the input to its eventual output. K-nearest neighbor, on the other hand, is a lazy learner that simply memorizes the training data and waits for test questions instead of creating a model to handle future instances. So you can imagine a take-home test where you can wait for the test questions and then pretty much control find the answer. Not that we would ever do that. However, anyway, the majority class of the neighbors then becomes the classification of the test sample. Support vector machines, or SVM, are seen actually a lot through biomedical literature, and I found them to be quite successful as well. Briefly, if you imagine these dots to be individual samples, you can see how there are a number of different decision boundaries. That is the boundary at which above would be red and below would be predicted to be green, can actually explain these data. However, support vector machine finds the support vectors or samples here that are most difficult to classify and then maximizes the distance from those hard to classify samples to determine its eventual decision boundary. And the last type of classifier I'm going to run through quickly is known as the multilayer perceptron. Now, the multilayer perceptron is a type of artificial neural network, like you may have heard from. And artificial neural networks essentially start with an input layer, run through one or more hidden layers in the middle, and converge onto a single output. Now, a multi-layer perceptron is one of these neural networks that is entirely interconnected. That is, each node is connected to each other. And these models, while they can be very successful, often tend to be extremely difficult to interpret. So it's almost described as a black box in computer science, and that you really don't know how it gets from its input to its output. But if the output is right, the output is right. And then moving forward with, like I mentioned a little bit ago, probe that memory retention metric as our outcome, our output, we must now figure out how we're going to evaluate these trained models. And there are a number of different ways you can train and validate machine learning models. The simplest way to do this is known as the train test split, where you literally split your data into, say, 80% training, 20% testing. Then you train the data with the training samples and assess the performance as a number of correct answers given in the testing phase. This is usually a little too simple for medical applications where our output can be very complex. So we turn to something called k-fold cross-validation. Now, k-fold cross-validation, you're going to separate your data into folds. Say I have 100 samples, I want 10 folds, and each fold will have 10 samples. And then set aside one fold for testing and train with the remaining 9. What makes k-fold cross-validation stronger is you are going to repeat this process until every single fold has acted as your testing fold. And then you report your performance, usually in the form of estimation statistics, as a mean average accuracy. And this is how most of my results are going to be shown. However, you can also report performance in a number of other ways. I'm not going to spend too much time here because I didn't apply them too much in my own research. However, they are very applicable for medical applications because using things like precision and recall is how we can actually identify a machine that is making a lot of false positives or a lot of false negatives. And you can start to imagine how you might actually take a less accurate model if it is completely avoiding the chance of making a false negative or missing a case of, say, cancer. So with that, a brief reminder, our first research question was given metrics from the first 7 days post-injury, including treatment. Can a machine accurately predict how an animal is going to be performing on that memory retention trial 30 days after injury? And in short, yes. You had the SVM performing at about 62% accuracy, which outperformed the baseline classifier at 48%. However, it's far from perfect. And while this does suggest that there are patterns in this data set that are allowing the machine to complete the task, they're not quite as striking as what we are looking for. So we really wanted to start picking apart why this might be. So we asked the question of, is including treatment as an input impacting our results? And to test this, we removed treatment as input and predetermined the folds to include only one treatment for testing and the rest for training. That is, the models that were trained on all but one of the treatments and then tested on samples that it had not seen yet. And this uncovered a huge amount of variability. And some were getting 0 and some were getting 100% in its ability to classify samples from treatments it had never seen before. And this kind of led us to start making the hypothesis that different treatments might actually be leading to different patterns of multivariate functional recovery. So we shifted focus a little bit, and to test this hypothesis, we turned our research question to, can a machine identify treatment administered when trained on a data set that includes metrics of recovery? That means our input is now metrics from the entire month post-injury, which includes physiology and biomarkers, cognition, and motor. And then the output becomes treatment class. When we did this with the FPI data, we were actually surprised to see that the machine was very, very good at it with near perfect accuracy in some cases. This means the machine was actually able to take the sample, clean, nicotinamide, etc. Got this one. This appeared from their treatment classes versus amantadine. the machine was quite bad at it and not really outperforming that baseline chance classifier in any of the situations. And then further, when we looked at the treatments relative to their vehicle controls, minocycline, which is pictured here, but the same for levetiracetam and EPO, were able to be identified pretty easily from their vehicles, while amantadine and nicotinamide were not. Once again, this is opposed to that less severe diffuse injury. And we were also surprised to see that the machine had great difficulty in distinguishing between doses of the same treatment. However, this may be less related to dose response and more related to the fact that OBTT, that original study, chose the doses by reported success in the literature and not by including a full range of efficacy or toxicity. So then we asked the question of what happens if we switch injury models? Will the machine still be able to accurately identify the same treatments as before? Will the results be the same for the treatments that were administered when we move from that FPI model to that CCI model? And while these data sets included common data elements across the board, they were not identical. So we had to return to that feature engineering drawing board. And the biggest change between these two data sets is that in the physiology for FPI, we actually had a lot of information about blood gases and blood pressure, things like that. Whereas in the CCI, we only have the weight, and then we calculated the percent weight change on surgery day versus 5 days after the operation. But we moved forward with the same set of classifiers and found that in all cases except for levetiracetam, the machine performed rather poorly. We didn't really see a whole lot of striking findings like we did with the FPI data. But levetiracetam actually performed quite well across the board. And this is the high dose pictured here, but it was also true for the low dose of levetiracetam. And looking forward at the rest of the comparisons, it only appeared to be true for levetiracetam. Even when we looked at the comparison to vehicles, it was really good at figuring out whether an animal was administered a lev or whether it was administered the other therapies. And so taken together, these results do confirm that pharmacotherapies are leading to unique recovery profiles following traumatic brain injury, and that these recovery profiles actually change depending on the type of injury that you received. And this represents a critical first step in identifying the treatments that might be optimal for a specific subgroup of patients. And we really wanted to know if our findings actually required machine learning, or if we could have gotten there with the typical analyses. So here we looked at the features and the feature groups through the application of PCA and repeating our analyses by removing one feature group at a time. And you can see that our principal components, which accounted for about 82% of the variance in our data set, were actually a large combination of the features involved instead of being overrun by one feature or two features. And you can actually see when we remove one feature group at a time, the accuracy loss is pretty equal across the board. So it wasn't like one feature or one feature group was driving these findings. It was really the combination that allowed us to get to these insights, suggesting that machine learning was actually necessary. And lastly, we're aware of the fact that binary classifiers are not really a good representation to the clinic where there are multiple treatment options. However, if we just start adding more output classes, so like I said, we were doing binary comparisons. If we tried to classify between 5, you can imagine how there would be a decrease in accuracy as we add more and more options. If there's 5 answers on a multiple choice test, it's harder to get the question right than if there's 2. And so to address this, we designed a novel machine learning architecture that builds off the strength of those binary classifiers, since we already knew they were very good at their job, and then generates an output of 2 features based on probability, and then ran a PCA on that redescribed data set, and then asked VM as a final estimator, a master classifier, to give us one treatment. So we asked it, out of these 5, which treatment did this sample receive? And you can see actually in cases of both 3 output classes and 5 output classes, this stacked architecture was much better than chance. However, we wanted to know if it was better than a simple classification. So this would just be chance, no information, versus an actual classifier that had, 5, could learn from the data set and had the 5 options, like I described a moment ago with the multiple choice test. And in the case of the 3-way classification, we saw that the stepwise classifier, that new architecture I just described, was actually about not much better, maybe 2% better or 4% better than the simple classifier. However, something that was really interesting was when we ran a dimensionality reduction on the output from that master classifier, we're actually now getting very distinct clusters by treatment class. And that is also true in the case of 5 treatments. Now we're seeing about a 12% increase in performance for the stepwise classifier versus that simple 5-way classification. And once again, we're starting to see much better clustering of the treatment options than we were at the beginning, or even from the combination of pairwise classifiers. So in conclusion, these techniques have really shown us their ability to generate novel insights, and that they've demonstrated that pharmacotherapies are leading to distinct patterns of multivariate recovery, that these patterns are differing based on the injury received, and that stacking architectures may really be a useful tool for us to help transfer our findings from bench to bedside. So with that, I would, of course, like to thank my fantastic mentors, as well as our collaborators and my dissertation committee for all of their advice and guidance along the way. And with that, if anyone has any questions.
Hi, Hannah. I have a question. Very nice talk. It's still very complicated for me, but I'm trying to learn a little as I go along. I had a question regarding dosing. So when you have all these drugs, say for example Keppra, and if you're looking at different doses, and your outcome shows that Keppra is beneficial, when we read this, how do we know what dose was driving force?
Well, in my analyses here, I separated them out by dose. I actually called levetiracetam Keppra high dose and Keppra low dose 2 different therapies and tested them that way. However, one important thing to realize is when I'm doing the analyses this way, it's not telling me anything about whether it's beneficial or whether it's deleterious. It's just telling me that it's different. And
Right.
that's kind of a way that I see these techniques as not necessarily a replacement for the typical analyses, but rather complementary. So Keppra in particular in the OBTT was actually the only therapy that was not deleterious in any injury model. So these findings kind of further confirm that this might be the way to go, because not only is it leading to univariate success, or at least not problems, it's leading to vast changes in the multivariate space.
Okay. I'll ask another comment. You had mentioned earlier, I think an interesting point about the novelty of this is that reviewers may not fully grasp it. And so you're going to get publications that may be crap, or they may be great, who knows? But the idea is when you submit this kind of work, are you concerned of who's going to read it? And maybe they'll be very excited by the idea and say, oh, great, accept as is. Or you have somebody who's like, I don't have a clue what they're saying, and just kill it right out.
Yeah, from our end, we tried to address the issue and make sure our work is good by having an interdisciplinary collaboration. But you're right, that's very difficult because there's not very many journals that have reviewers that are just as interdisciplinary as our collaboration. And, we run into that sometimes where you can definitely see who has more informed questions than others. But we really do our best to find a common language that can explain these techniques in a way that kind of goes at that intersection of computer science and brain injury, so that not only can we get our message across, it also gives the reviewer enough background to, if they don't understand something, look up the relevant information.
Well, I think there's a lot of potential here, so good luck.
Thank you. I hope so.
I have a question.
Oh, hi, Dr. Bramlett.
Hi. So, great job. Good talk. So my question is, what's your next step? Now that you've analyzed the FPI data and the CCI data, I know you didn't have histology. So what are you going to do next with the machine learning paradigm and the OBTT?
Well, with some of this work that we've done at the end, one of our goals is to develop something that is able to be informative in the clinic as well. So I've actually been working on figuring out how to best analyze imaging data. So maybe I can take a lot of what I've learned to a clinical data set and see if the findings that we've discovered with OBTT actually hold true in the clinical arena as well. But right now I'm focused on getting these results published and hopefully graduating from my PhD.
I guess if that's what you want to do, that's fine. But yeah, no, obviously, yeah. So moving to the clinic is a big next step, because that's what we want to do. So in terms of predicting outcomes and stuff. But anyway, thank you. Good job.
All right, I'm going to go ahead and close the room. If anyone has any questions for Hanna, please reach out to her via email, or you can reach out to me and I'll go ahead and forward it on to her.
Great job, Hanna.
Thank you.